Okay, so welcome back from the break. Let's take a look at our concept check questions before we move on. So why is genetic variation in a population a requirement for evolution? Well, what you have to remember is that it is phenotypic variation that natural selection works on. That's the raw material for natural selection. And phenotypic variation is only going to occur if there's an underlying genetic variation. So in the absence of genetic variation, say for instance, there is very little heterozygosity. Remember, that's what we measure to measure the level of genetic variation. If there's very little heterozygosity, in other words, at any particular loci or in the sum total of all the loci, the physical locations of the genes in the chromosomes, there is only a single allele, which we would, and the terminology that we use for that is to say that this particular allele is locked at that loci. There's no other alternative. Then there would be no genetic variation, which would result in no phenotypic variation, which would lead to a loss of natural selection, and there would be no evolution. So having genetic variation that leads to phenotypic variation is central to the possibility for evolution, because that is what allows for natural selection. In the absence of natural selection, there's no evolution. Not in this case, at least, with genetic variation. Mutations occur spontaneously in populations all the time. Why do only a small number become widespread in a population? Well, remember, the vast majority of mutations are going to occur in somatic cells. Somatic cells are not going to make any contribution to the next generation because they do not give rise to eggs and sperm. So the vast majority of mutations there simply have no effect. So we can forget all of those. The only mutations that could potentially have an effect are those that occur in the germ cells. Of those mutations, most are going to be either neutral, some are going to be lethal, which means they will get eliminated from the population very quickly because of their lethal nature. Only a small number are going to be beneficial, and only those will have the potential to become more widespread in the population due to sexual reproduction and natural selection. So unless the mutations occur in the germ cells and are specifically beneficial at that point in time and at that physical location, and therefore favored by natural selection, they're not going to become widespread in a population. They're simply going to be lost. So that's the reason why only a small number of mutations become widespread in any population. It's the limitations of natural selection and the limitations of sexual reproduction, as powerful as they may be. So let's move on and continue talking about population genetics. So one of the important tools for us as biologists interested in population genetics is what has become known as the Hardy-Weinberg theorem. Hardy was an English mathematician who was not actually a scientist at all. And the only contribution that he ever made to biology was here in the Hardy-Weinberg theorem. Weinberg was actually a German biologist who independently and at roughly the same time came up with the same idea that Hardy developed. Now, I have a pretty good idea about how Hardy and why Hardy developed his particular set of mathematics, which I'll show you shortly. I'll actually run through the derivation of the formula, although basically some of that legwork will be done in the next couple of slides, but I'll go through the formal derivation of the formula shortly, as I believe Hardy demonstrated it. Both Hardy and Weinberg sent papers in to the same journal, Nature, at roughly the same time. And when the editor read both of the papers, he realized that they were essentially stating the same thing. So he contacted both Hardy and Weinberg and asked if he could have their permission to publish the two papers together. And so the two papers were published together in the same journal, and both Hardy and Weinberg were given equal credit for the idea. Consequently, this became known as the Hardy-Weinberg theorem. And what they demonstrated mathematically was the conditions under which a population would not evolve. And what that implies is that there is no change in allele frequency over in this population from one generation to the next, that the frequency of the alleles that these populations possess, or a population possesses, remains constant from one generation to the next. And the most important feature of the theorem is that the only mechanism for creating, I guess, the allele frequency from one generation to the next is Mendel's principles, segregation and independent assortment are the only two processes that would work. In other words, the processes of meiosis. And if those are the only two processes that are at work, Mendel's principles imply that the frequency of alleles from one generation to the next must remain constant. Now, as you'll see a little bit later, there are 5 assumptions that Hardy and Weinberg realized that you had to make in order for this to work. In order for Mendelian genetics to be the only process at work in distributing alleles from one generation to the next as a consequence of meiosis. But let's take a look at what this looks like statistically in terms of numbers, because it's easy to see why Mendelian inheritance maintains the frequency of alleles by doing basically a Punnett square analysis. So if we have two populations in which there are 2 alleles for a gene, again, Mendelian principles imply that there are only 2 alleles. So let me fire up my pen for a moment. So it's Mendelian genetics. And the implication of that is only 2 alleles. Now, let me just add on to that of any gene, because that was the one thing that Mendel stated. And the reason why this was based on Mendelian genetics was because at the time, Mendel's genetics principles were the only genetics principles that were widely understood and widely accepted. Morgan's work had not yet been published. Other work that you might be familiar with from Bio 1 had not been published yet. So the only principles that were in play when Hardy and Weinberg were developing their concept were the principles of Mendel. So we only work with 2 alleles. So here we have a population of organisms that has 2 alleles for a particular gene. One of the alleles is present at a frequency of 0.8. In other words, 80% of the alleles are the CR allele. The other 20% are the CW allele. And because there are only these 2 alleles, obviously this adds up to 100% of all the alleles. So the frequency is given as a decimal value. So the frequency of the CR allele is 0.8. The frequency of the CW allele is 0.2. So when those individuals in this population reproduce, the gametes that they produce, because those the genes segregate into the gametes randomly, again, part of Mendel's inheritance principles. This is the law of segregation. The frequency of each type of allele in the gametes should reflect the frequency of the alleles in the population. So given that 80% of the alleles are CR, 80% of the gametes, whether they be eggs or sperm, should contain the CR allele. the other 20% of gametes, given that the CW allele is 20% of the alleles in the population, should represent that. And so 20% of the gametes should have the CW allele. If we then apply the Punnett square to this, and we use heterozygous individuals where there is going to be alternative outcomes, so we focus on heterozygotes because they have alternative outcomes. Being heterozygous, they can either supply the CR allele or the CW allele. Homozygotes are not going to affect the outcome because they are only ever going to pass on the one allele that they have. So those that are homozygous for red would pass on just the CR allele. Those that are homozygous for white are only going to pass on the CW allele. That's all they can pass on. And so in terms of their effect on the final population, the frequency or the probability of them passing on a CR in the case of the homozygous for red is an absolute 100%. That's a frequency of 1. And remember, in mathematics, 1 times a number is that number itself, so it could change anything. Likewise, we make the same argument for those that are homozygous for CW. So the only parental types, the only genotype that can really have an influence on the next next generation are those that provide alternatives. Those are the heterozygous individuals. So that's why we look at the heterozygous in the Punnett square. So here are the heterozygous eggs and sperm. We have CR eggs and CR sperm. And if those two come together, we get a red individual. They're homozygous for the CR allele and they're going to be red. And their frequency is going to be the product of the frequency of these two alleles. So it's 0.8 squared. That gives us a frequency of 0.64. So 64% of the individuals are going to be homozygous red. For the CWs, which are down here, they're going to show the white phenotype. They're homozygous white. And again, we multiply the two frequencies together. That's 0.2. 0.2 squared gives us a value of 0.4. Oh, sorry, 0.04. That's 4% of the population. Then we have two groups that are going to be heterozygous, each representing 0.16 because in terms of frequency, because that's 0.8 by 0.2. So this is going to be 16%. And this is going to be 16%. But in terms of overall, that's going to be a total of 32% of the offspring. So when we want to figure out the frequency of the alleles in this population, we've got to take into account both the frequency of the individuals in the population. Again, remember, this is at random, this is the random distribution, but we've also got to take into account whether they're homozygous or heterozygous. So if we're looking at the CR allele, we know that all the individuals that are homozygous red or homozygous dominant have 2 copies of the CR allele. So all of the alleles in the red individuals are the CR alleles, and that constitutes 64% of the alleles are going to be CR from these individuals. But that's not the only source of the red allele, because remember, all the heterozygotes also have the CR allele, but they only have 1 copy. So even though 32% of the population are CRCW, only half of their alleles are CR. So we don't use 32%, we use 16%, half of the alleles. So for the CR alleles, it's 64% plus 16%, well, 16 plus 64 is 80%. And remember 80% reduces to 0.8 as a probability or as a frequency number, which is exactly the same as what it was previously. For the CW allele, again, all the homozygous individuals have two copies of the CW allele. So we take all 4% as being the CW alleles because of these homozygous individuals. And again, same argument for the heterozygotes. Half of the alleles in the heterozygotes CW. So that's 16%. 4 plus 16 is 20%, which comes to 0.2. And these two numbers are exactly the same as they were before. And of course, you can, it's not a very big leap to realize that if this generation breeds again randomly, based on these frequencies, it should be repeat. It should just repeat. And so long as there is no change in the frequency of these alleles in the population, there's not going to be any change from one generation to the next. In other words, the information, the frequency of the alleles is not going to change, therefore the population is not going to evolve because it requires a change in the frequency of the alleles in the gene pool in order for the population to evolve. So long as Mendelian genetics, Mendelian inheritance principles, are the only principles in play for the distribution of alleles from one generation to the next, the population is going to be in equilibrium, what we refer to as Hardy-Weinberg equilibrium. So what's the actual formulas that Hardy and Weinberg generated to indicate or to demonstrate that the populations were actually in equilibrium under particular circumstances? Well, it came from a conversation that the British geneticist Punnett, he of Punnett Square fame, had with Hardy because they were faculty members at the same university and they encountered each other on a fairly regular basis at the faculty cafeteria. And Punnett had read a paper from a German biologist claiming that the only circumstance under which a population would remain static would be if the two alleles, and again, remember, Mendelian genetics is in play at the moment, but the only circumstance under which a population would remain static is if the two alleles were present in a 1-to-1 ratio. In other words, 50% of the alleles were the dominant trait and 50% of the alleles were for the recessive trait. Punnett realized instinctively that this was not correct, that there had to be other conditions under which the population would remain static. But he also realized that he did not have the mathematical tools to demonstrate that. He just knew instinctively that it could not be correct, that there would be this one situation. So when he next encountered Hardy, he explained his problem to Hardy, and Hardy sat down for half an hour in the cafeteria writing on the back of a paper napkin what became known as the Hardy-Weinberg equations, because Weinberg developed exactly the same set of equations independent from Hardy. I have a pretty good idea about how Hardy went about doing it, so it's probably pretty much the same for Weinberg as well. So the next slide is a blank slide. You're not going to see this slide in the slides in the PDF right now, but I will substitute in this so that you, well, actually I can't because I don't save these changes because I want to keep this slide set pure. But you can take a look at this derivation, and it's not really important that you remember the derivation. I just want to show you how you arrive at the formula so that you understand a little bit better what the formulas are actually telling you and why they apply. So we have to remember that this is based on on Mendelian principles. So we have to start with Mendelian principles. And the most important here is only two alleles, which for our purposes we will call big A and little a. So given that those are the two alleles that are available for a particular gene, it stands to reason that the sum total of those two alleles in the population must come to 100% because they are the only two alleles present. So the percentage of big A plus the percentage of little a must equal 100%. Or to put it in probability terms or frequency terms, big A plus little a must equal 1. 1 is a certainty, 100%. So when we start thinking in terms of possible genotypes then in a population as a consequence of these two alleles, we know that there are 3 possible genotypes. Big A, big A, big A, little a, and little a, little a. Now, as I explained in the previous example, the ones that are of most importance to us here are the big A, little a individuals, the heterozygous individuals, because they are the only ones that provide alternatives. So in terms of the influence on the next generation, it's only the heterozygous individuals that are going to have any influence, because the outcome from the homozygous dominant, big A, and the outcome from the homozygous recessive, little a, is a certainty. They can only ever pass on the big A in the case of the homozygous dominant, or the little a in the case of the homozygous recessive. Only the heterozygotes can pass on the alternatives, a big A or a little a. So the key cross here for the next generation is the big A, little a, cross big A, little a. Now, when we think in terms, however, of the alleles that these individuals can pass on, yes, these individuals can pass on either a little a or a big A, but they must pass on one of them. So their contribution to the next generation has to be a big A or a little a, but it has to be one of the two. So their total combination in terms of the percentage alleles that they're going to pass on to the next generation is the sum total of the percentage of big A they pass on plus the sum total of the percentage of little a that they pass on. Big A plus little a. Well, we already know what that is. Big A plus little a adds up to 1. Crossed with, again, same argument. This individual must be in the same position. Now, this then becomes distributive multiplication, because if big A plus little a equals 1, big A plus little a multiplied by big A plus little a must also equal 1, mathematically. And now it's just an algebraic equation involving distributive multiplication. So we multiply big A by big A, and let me just move this out of the way for a moment. So we multiply big A by big A, and that gives us big A or big A squared, plus big A multiplied by little a, which is big A little a, plus big A multiplied by little a, plus little a multiplied by little a, or little a squared, and that must all equal 1. But now we can reduce that equation a little bit further, and it becomes big A plus 2 big A plus little a equals 1. But now when we look at this and we look back at our 3 genotypes, we see our 3 genotypes represented here. Our homozygous dominant, our heterozygous, and our homozygous recessive. And the frequencies of these 3 genotypes in a population must all add up to 1, which stands to reason. And so this equation now helps us get a handle on the frequency of the 3 genotypes in a population, because each of these terms represents the frequency of one of the 3 genotypes in the population, and the sum total of the 3 genotypes must add up to 1. Likewise, this equation up here tells us that the frequency of the 2 alleles for any particular gene must also add up to 1. And so long as we know the frequency of one allele, or we know the frequency of one of the 3 genotypes in the population, we can figure out everything else. And the one particular individual in the population that we really like to know, and the one that we can usually get a handle on, is the homozygous recessive. Because if a gene is recessive, or if a trait is recessive, for an individual to show that trait, they must be homozygous recessive. We know that absolutely. If an individual shows the dominant trait, we don't know whether they're homozygous for it or whether they're heterozygous for it. But we do know that an individual showing the recessive trait has to be homozygous for it. So we can generally get from any population the frequency of the individual showing the homozygous recessive trait. From that, we can calculate the frequency of the recessive allele, what we're referring to as A here, but as you'll see, we use a different symbol. Once we know little a, we can very quickly figure out big A, because big A is simply 1 minus little a. And then once we know big A and little a, we can figure out big A, and we can figure out big A, little a, and multiply that by 2 and figure out how many heterozygous individuals there are. So this is how Hardy and Weinberg independently would have figured out their 2 equations. The only difference is that today, rather than using big A and little a, we use P to represent the frequency of the homozygous allele. And it's really important that you remember what each of these symbols represents, because the one thing that I find from one semester to the next that really trips up every student who encounters the Hardy-Weinberg equation is remembering what each of the symbols refers to and how to relate that to the data that you might be given in one of the Hardy-Weinberg problem. Now, I'll show you how to work a few problems in this presentation. In the population genetics lab, you're going to be reintroduced to Hardy-Weinberg, and you're going to be practicing multiple examples of these calculations, so that you are going to get many opportunities to practice these calculations, because ultimately in exam 1, you're going to be working a couple of these problems. And it's really important that you master this. So P is the frequency of big A, Q equals the frequency of little a. So it's the frequency of the dominant, the allele for the dominant trait and the allele for the recessive trait. P squared is the frequency of big A, homozygous dominant. 2PQ is the frequency of the heterozygotes, big A, little a, and Q squared is the frequency of little a, or the homozygous recessives. So P is the frequency of the dominant trait in the population, or in the population. Q is the frequency of the recessive trait, or sorry, the recessive allele. So let me back up and make sure I correct this, because I have given you something that's incorrect. P is the frequency of the allele for the dominant trait. So it's the dominant allele, if you want to use that terminology. I don't like that because it's not correct, it's not accurate. It's the trait, it's the allele for the dominant trait. Traits are dominant and recessive, alleles never are. Alleles are just alleles. The traits are what are dominant and recessive. So big A is the allele for the dominant trait, and P is the frequency of that allele in the population. Q represents the frequency of the allele for the recessive trait in the population. P squared is the individuals who are homozygous dominant. 2PQ are the individuals that are heterozygous. Q squared are the individuals who are homozygous recessive. So P and Q refer to the alleles P squared, Q squared, and 2PQ refer to that refer to individuals. And it's really important that you remember this distinction, because it's so very easy to confuse these. So you may recall that I said a little earlier in the previous section that there are assumptions that have to be made in order for Mendelian genetics to be the only principle that's in play here. So here are the 5 assumptions or conditions that underpin the Hardy-Weinberg theorem. And this is what we're really interested in as biologists. Yeah, mathematically, it's really interesting to play with the Hardy-Weinberg theorem. But in this class, we're not functioning as population geneticists. This is not a population genetics class. So our interest in these assumptions is a little bit more functional, rather than being used for underpinning the theorem. Because once we understand what these 5 conditions are, we also understand what we should be looking for to identify a species that is evolving, or a population that is evolving. Because any evolving population is going to break one or more of these assumptions. So the first assumption is that the population should be large, because remember, this is all about statistics. It's all about frequencies and probabilities. And statistics works better with big numbers, not with small numbers. And so the first assumption is that the population is very large, ideally infinitely large. Obviously, that's not possible. But the larger the population, the more likely it is that it could reach Hardy-Weinberg equilibrium. There should be no gene flow. That is, individuals should not be moving between populations. That's why recognizing that in those previous examples of overlapping ranges of adjacent populations versus where there is exchange of alleles versus no exchange or very little exchange between overlapping ranges of populations is important, because if there is an overlap of the range, there is the potential for exchange of alleles between the two populations due to migration of individuals. Because when individuals leave a population, they take their alleles with them, which automatically changes the frequency of those alleles in the population that they're leaving. When they enter a new population, they bring their alleles with them, which again automatically changes the frequency of those alleles in the new population. And so in order for the allele frequencies not to change, there has to be no migration at all. There should be no mutations, because remember, mutations are the source of alleles, new alleles. So if you don't want to change the allele frequency, and that automatically changes with the appearance of a new allele, then there should be no mutations. And of course, logically, we realize that that's simply not possible. But nonetheless, it is one of the requirements for a population to be in Hardy-Weinberg equilibrium. There should be truly random mating, because if you recall our statistical model, the CR-CW model, the only way that the CR and CW alleles could turn up in the same frequencies in the next generation was if each of those alleles occurred in the gametes randomly at the same frequencies as they occurred in the population. And that can only happen if mating is truly random. If all individuals have equal opportunities to mate, and therefore equal opportunities to pass their alleles on to the subsequent generation. And of course, we know that natural selection must also be ruled out, because natural selection requires differential reproductive success. In other words, some individuals leave behind more offspring than others. So random mating differs from natural selection in that random mating means that all individuals have an equal opportunity to mate. That means they must all survive to reproductive age. There must be no differential survival. And then natural selection, because in natural selection there is the implication that those individuals that are better adapted are the ones that are more likely to thrive to mating age, and therefore more likely to leave behind more offspring. Differential reproduction, natural selection has to be ruled out as well, because that would eliminate the opportunity for some individuals to mate. All individuals must not only have an equal opportunity to mate, but equal opportunities to leave behind equal numbers of offspring. So what sort of things would we be looking for? Populations evolve if one, small size. And we'll talk more about that. I'm just looking at this and I just realized that I've got a typo. It should say that there is gene flow and therefore migration. And my whole explanation of that was wrong. Yeah, I do make mistakes, and I'll be the first to admit to it. And I'm not sure why I didn't even realize this before. I know I put myself up short on this in previous classes, and I'm pulling myself up short on this one again, because I just realized just exactly what's so wrong about this. And I should have realized this when I was talking about it as to why it's wrong. And I'm not sure why I didn't. So I'm making an admission here. I made a mistake. Make sure you don't make the same mistake that I made. The reason why gene flow is important, the reason why migration is important, I'm rethinking. I'm thinking about this thing again. I was right the first time. And I was right the first time for a reason, but there is something sitting in the back of my mind that I'm going to have to explain to you a little bit later on in the slide. It's not that I'm wrong. It's that there is something that we'll touch on in the future, both in this chapter and an upcoming chapter, where gene flow does serve a very real purpose in preventing evolution, preventing a population from evolving. We'll talk about that in a minute. We will talk about that in the future. So, and I may have to go back and think about this and do a little bit more reading, because yeah, I'm really stuck on this one right now. So those of you who are listening and watching this, I may have to send out an email explanation on this one, because I'm really struggling on this, and I don't want to sit here talking about this any longer, because this presentation is going to be long enough, and I'm kind of time limited today as I'm making this. So I want to keep moving. So gene flow, obviously movement of individuals from one population to another, does change the allele frequency. So a population will evolve if there is migration, but I want to highlight on that. So migration occurs. Number 3, if there's mutation, which there always is. Number 4, differential mating. Not everyone gets the same mating opportunity. Or actually, yeah, differential mating. And number 5 is differential survival. Now, I'm in the back of my mind, I'm still going over the issue with the no gene flow. I'm going to have to revisit that one and issue a longer note on this one. I can see that. I need to resolve this one. You've got, for the first time in a while, I've been forced to really stop and think about this one. Anyway, let's keep moving. So there is always a hypothetical possibility for a Hardy-Weinberg population. In other words, a population that is in Hardy-Weinberg equilibrium. So it's always hypothetically possible. But in the real world, absolutely not, because we know that mutations always occur and there's no way of stopping that. So right there, that assumption is automatically broken, which means populations always have the possibility to evolve. So in terms of human genetics, there are individuals, and you may have heard about these in your Bio 1 class, that work as genetic counselors. In fact, we have a former genetic counselor as a part-time faculty member for us, teaching biology classes. And genetic counselors use what we know from the Hardy-Weinberg equation and use the calculations to figure out the possibility of individuals randomly possessing an allele for a particular genetic condition. This is a really important calculation for individuals who know that there's a family history of a particular allele in their family gene pool. So a condition that you may have encountered in Biology 1 is called phenylketonuria, otherwise known as PKU. This is the condition where an individual is unable to metabolize the amino acid phenylalanine correctly. It's the reason why there are warnings on diet sodas that contain aspartame or NutraSweet for phenylketonuria, people who have this condition, because aspartame is actually a dipeptide, two amino acids that are linked together in such a way that they can bind to the sweet taste buds in your mouth, and you detect it as being sweet. But one of the two amino acids happens to be phenylalanine. And because if it is left untreated, if an individual is exposed to phenylalanine from birth, it is the most common cause of delayed mental development. And it can be easily prevented by simply not allowing an individual to be exposed to phenylalanine, or at least reducing the exposure. And so for individuals who are identified as being PKU positive, and there is a genetic screening that is mandatory in all 50 states. So if you are under the age of about 30, you've actually been screened for this. And it is mandatory that you be screened within 48 hours of birth. It's a simple blood test. But you can calculate the probability of any two parents randomly possessing this allele, in other words, being heterozygous for it. Now remember, heterozygosity, or the heterozygous individuals, are the 2PQ individuals. So if we know nothing about the frequency of the allele in the population, that doesn't matter, because PKU happens to be a recessive trait. So assuming that the frequency of PKU individuals, those possessing the recessive trait and therefore being homozygous recessive, is 1 in 10,000, that gives us a decimal of 0.0001. Now from the p squared plus 2pq plus q squared equation, q squared represents the homozygous recessive. Pen's not active. Let's bring that up. So remember, this is a recessive trait. And this is usually the data that you have. You'll be given a birth frequency or birth rate of individuals with the recessive trait. And so that automatically gives you Q squared. Now be careful, because some genetics problems don't necessarily give you that number. So don't automatically assume that the data point that you've got represents the homozygous recessive. Read the question carefully, make sure you understand what the data represents. But in this case, 1 in 10,000 represents the birth rate for individuals who are homozygous recessive, because PKU happens to be a recessive trait. So Q squared automatically equals 0.0001. So Q, the frequency of the PKU allele in the population, represents the square root of Q squared. So Q equals the square root of Q squared, which equals 0.01. Therefore, P equals 1 minus Q, which equals 0.99. Now, what we want is not P squared or Q squared, not P squared, which we could calculate if we wanted to. What we want is the value for 2PQ. So 2PQ equals 2 times P, which is 0.99, multiplied by Q, which is 0.01, and that all comes out to a value of 0.0198, or approximately 2%. So at random, 2 out of every 100 individuals, or 1 out of 50 individuals, is heterozygous for the PKU allele, just randomly. Now, you may think that sounds like a really high incidence. It's not actually. It's actually quite a low incidence. Some alleles, like the sickle cell allele, again, a recessive trait, occur at a frequency in African American populations of around 1 in 12, I think it is, 1 in 12, 1 in 15 individuals are heterozygous for the sickle cell allele. That's a high frequency rate compared to 1 in 50. And we might address that a little bit later, depending on how we're going for time, because we're going to talk about the reason why the sickle cell allele exists in the first place. But let's move on. So another quick break with some concept check questions. So these are Hardy-Weinberg questions. What are the frequencies of the alleles for big A and little a in a population of 700 individuals? If 85 are homozygous dominant, 320 are heterozygous, and 295 are homozygous. And number 2, in a population at equilibrium, the frequency of the little a allele is 0.45. What are the calculated frequencies of the big A, big A, little a, and little a genotypes? See you on the other side of the break to do the calculations.